that is the most important part in botany, which is known as molecular biology. Let's start with the first portion, that is known as nucleic acid. The nucleic acid is an acid which is mainly composed of the acid called phosphoric acid or H3PO4. The mainly acid. Then let us know that who discovered nucleic acid, who coined the term nucleic acid, then the type of nucleic acid, and then the different part. Let's start know who coined the term, who discovered nucleic acid. The scientist Frederick Mister discovered nucleic acid, and then the scientist Altman coined the term nucleic acid. Then let's move on to the next part, which is the type of nucleic acid. There are mainly two types of nucleic acid. One part is known as DNA and another part is known as RNA. The DNA is otherwise known as deoxyribonucleic acid, and the RNA is otherwise known as ribonucleic acid. Then there are mainly two types of nucleic acid. One is DNA and another is RNA. Then let's move on to what is the nucleic acid. The nucleic acid is a chain of polymer of nucleotide. What is nucleic acid? It is a chain of polymer of nucleotide. Then what is nucleotide? Then the nucleotide is known as, it is the mainly the composition of nitrogenous base, pentose sugar and phosphate group. When the three part combine together to form a nucleotide, the first one is nitrogenous base, then the second one is pentose sugar, and the third one is phosphate group. If we combine with that the nitrogenous base and the pentose sugar, then it is known as nucleoside. Then what is nucleoside? It is the composition of nitrogenous base and pentose sugar. If we will add with phosphate group, then it is known as nucleotide, and minus phosphate group, it is known as nucleoside, which is known as nitrogenous base and pentose sugar. Then let's move on to the next part, which is known as nitrogenous base. The nitrogenous base, there are mainly two types of nitrogenous base. One is purine base and another is pyrimidine base. There are mainly five nitrogenous base are seen. There are mainly two types. One is purine and another is pyrimidine. In the purine base, the first one is adenine, second one is guanine, and in the pyrimidine base, first one is thymine, next one is uracil, and the last one is cytosine. There are mainly five types of nitrogenous base in adenine, guanine, thymine, uracil and cytosine. Next, move on to the pentose sugar. What is known as pentose sugar? Pentose sugar is a 5-carbon compound. That is a sugar which contains a 5-carbon compound. The formula is C5H10O5. Then there are mainly two types of pentose sugar. One is ribose pentose sugar and another is deoxyribose pentose sugar. The ribose pentose sugar contains one more oxygen than the deoxyribose sugar. The main formula is C5H10O5. Let's make the graph. C5, C1, 2, 3, 4, 5. Carbon 1, 2, 3, 4, 5. Then there are mainly two types of pentose sugar. One is ribose sugar and another one is deoxyribose sugar. Then then the last part is phosphate group. What is phosphate group? Now it is the component of an acid which is mainly known as phosphoric acid, which is H3PO4. Then this group is known as phosphate group. The part of a phosphoric acid is known as phosphate group. Then there are mainly two types of nucleic acid. First one is DNA, next one is RNA. Then the nucleic acid is a polymer of nucleotide. Then what is nucleotide? Nucleotide is the by the chain of nitrogenous base, pentose sugar and phosphate group. Then what is nitrogenous base? There are two types of nitrogenous base. One is purine base, another is pyrimidine base. The purine base are adenine, guanine, and the pyrimidine base are thymine, uracil and cytosine. Then there are mainly two types of pentose sugar. One is ribose pentose sugar, another one is deoxyribose pentose sugar. There are mainly two types of pentose sugar, and the last one is a phosphate group, which is a combination of phosphoric acid or H3PO4. Next portion, what is DNA? DNA is a deoxyribonucleic acid. Then let's start with a major part, which is known as structure of bDNA. The structure of bDNA, first the scientists who discovered the structure of bDNA, that is Watson and Crick Franklin, discovered the structure of bDNA. Then mainly DNA, it is a deoxyribonucleic acid, which is which is mainly a two strand, but which is there are mainly two strands in the deoxyribonucleic acid. Then we have to know the structure of DNA, then the loop which shows phosphodiester linkage, then the significance of the conclusion of bDNA. Then first we have to know what is the bDNA. The DNA is a deoxyribonucleic acid, which is mainly having one less oxygen atom. In the first, there are mainly two polynucleotide chain, and the interior is one loop. Then there are mainly two end, one is 5' end and another one is 3' end. One strand is parallel to another strand, and the next in the other part it is anti-parallel to each other. The gap between these is 20 angstrom, and the gap between the loop is 10 angstrom. And let then describe the major nitrogenous base found in DNA, that is mainly ATGC, adenine, thymine, guanine and cytosine. In the portion of RNA, thymine is replaced by uracil. Then there are mainly four types of nitrogenous bases, A, T, G, C. Then there is in the purine base that A is linked with T by double bond and G is linked with C by triple bond. Then let join them together, that is A linked with T, G linked with C, A linked with T, A linked with T and G linked with C. In the process there is the linkage occur. Then it is a very easy visible that a loop, this portion which is very clearly visible, that there are mainly two ends, one is 5', 3' to 5', another one is 3' to 5'. Then one is, this is pentose sugar and this is phosphate group, and this is the nitrogenous base. The nitrogenous base, the pentose sugar, and this is the phosphate group, in which linked each other. The two nitrogenous base linked with third carbon and with fifth phosphate group. Then it is known as always 3. Then the next part is G react with C, in which G it is a triple bond. Then it is react with the pentose sugar and the phosphate group, in which then the next part A react with T, A react with T and G react with C. Then there is also, if one strand is like this, A, G, C, T, then the next strand will be A, T, G, C, C, G, A. That is one is known as complementary strand. So if one strand occur in that, then the opposite strand will occur in the other. There is a very common DNA which is seen in that is known as, if we will take in a mirror image, that is G A A T C. If we will reverse it, C T T A A G. That is exact opposite, which is known as palindromic DNA. Then there is there are the type of DNA. One is aDNA, another one is bDNA, another one is cDNA, another one is dDNA, another one is eDNA, another one is zDNA. There are 11 base pair in aDNA, 10 base pair in bDNA. In cDNA there are 9 base pair. In dDNA there are 8 base pair, and in eDNA there are 7.5 base pair, and in zDNA which is 12 base pair. This structure is mainly of bDNA, which is clearly visible. And the DNA or the deoxyribonucleic acid is mainly used for the cell division. It is mainly used for the hereditary purposes and mainly in our day-to-day life. This is all about your structure of bDNA. portion which is known as RNA. In the last part we have discovered about DNA, structure of DNA, function of DNA. In this portion we have to discover about RNA. Then what is RNA? Types of RNA. Then the different part. What is RNA? It is a single stranded structure. It is a single stranded structure in which there is only one polynucleotide chain. Then there are mainly three type of RNA. One is rRNA, next one is mRNA, and the last one is tRNA. First rRNA, which is otherwise known as ribosomal RNA. rRNA is otherwise known as ribosomal RNA. In the rRNA, there is 80% of the total composition of the cell. In rRNA, there are mainly about 450 to 55,000 nucleotides and 25,000 to 10 lakh polynucleotide chain. In the rRNA, there are mainly 450 to 55,000 nucleotides and 25,000 to 10 lakh polynucleotide chain. This is all about your rRNA, which is also known as ribosomal RNA. Then the next part, it is the major part, that is known as tRNA, but transfer RNA, in which it carries the data to different part, that is known as transfer RNA. It is mainly consist of about 10 to 20% of the total number of cells. Then there are mainly the 75 to 100 nucleotide, and the next there are mainly 25,000 to 35,000 polynucleotide chain. Then the tRNA is also otherwise known as clover leaf model, in which it is given by the scientist Clover. This is the structure which is known as clover leaf model. There are mainly 4 loop: DHU loop, T-C loop, S region, and anticodon loop. In the DHU loop, it is also known as dihydroxyuracil loop, in which the tRNA is react with the amino acyl synthetase. In the T-C loop, it is known as the ribosome binding site, and the S region, which is variable, now which can be constant, and mainly it is react with the CCA end. But in the 3' portion, it is react with the CCA end. In the tRNA, the translation mechanism occur. It is the carrier. Then this is it is all about your tRNA, in which it is known as transformal RNA, in which there is 10 to 20% of total number of cells, in which there are 75 to 100 nucleotide, in which there are 25,000 to 35,000 polynucleotide chain. This is otherwise known as clover leaf model. Then it is the structure of clover leaf model. Then there are mainly 4 parts: DHU loop, T-C loop, S region, and anticodon loop. In the DHU loop, it is also known as dihydroxyuracil loop, in which it is react with the amino acyl synthetase. In the S region is variable, which can be constant at a particular region. The T-C loop is the ribosomal binding site, and it is the anticodon loop. It is bind with the CCA end, that is cytosine, and adenine end, that is known as CCA end in the 3' end. This is all about your rRNA and tRNA. In the last part, it is known as mRNA, which is mainly discovered by the scientist Jacob and Monod. It is mainly consist of 3 to 5% of total number of cells. There are mainly 900 to 12,000 nucleotides, and the total number of polynucleotide chain is 23,000 to 10 lakh. This is all about your RNA. And mainly there are 3 types of RNA: rRNA, mRNA, and tRNA. rRNA is ribosomal RNA, tRNA is transformal RNA, and mRNA is messenger RNA. This is all about your DNA, RNA, and RNA form. Thank you.